Hello, everyone, and welcome to today's webinar, NGS Library Prep and Quantification on the OT-2. Today's seminar is presented by LabRoots and brought to you by Opentrons. To learn more, visit opentrons.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble hearing or seeing the presentation. I'm Kinnery Watson, a director at Opentrons, and I'll be your moderator for today's event. Today's speaker will be Matt Akana, lead NGS scientist at Opentrons. He'll be presenting data he has produced on automating NGS library prep on the OT-2, as well as quantification, normalization, and pooling of NGS libraries. Before we start the presentation, I want to introduce you to the OT-2. The OT-2 is a liquid handling robot that's different from other liquid handlers. It was designed to be accessible to any life scientist. It's highly accurate, but extremely affordable and small enough to fit on half a lab bench. It's flexible and open, so you can add different pipettes and modules depending on the application, and you can use your existing reagents and labware. It's accessible to users with and without automation experience with a user-friendly GUI protocol designer and an advanced Python API. We offer support from free onboarding to custom protocol development and technical support. And you can automate a wide range of applications on this platform. Some of the most common applications are NGS library prep and quantification, PCR setup, nucleic acid isolation and purification, small-scale protein purification, biochemical assays, as well as general liquid handling procedures, including serial dilution and cherry picking, microplate and tube filling, as well as normalization and sample preparation. Matthew, you may now begin your presentation.
All right, thank you for that introduction, and let's get started. So first I'm going to go over an agenda of some of the things I'll be talking about in this presentation. So first of all is the basics of NGS workflow. That is next generation sequencing. In particular, what I'm talking about is sequencing on an Illumina-based sequencer. Then going into some more in-depth about library prep on the OT-2, what it is we've done so far, and how do we approach doing that using an OT-2. And then taking some time to talk about quantification, normalization, and pooling. These are steps that are crucial to NGS, but are not really elaborated too much on in the protocols for your library prep. A lot of this stuff is sort of left for the user to figure out on their own, and a lot of these solutions are particular to each individual lab. So how do we tackle some of these things using the OT-2? Basics of the NGS workflow. This is a workflow, and you'll be seeing it again as I refer to it in my presentation. These are sort of the steps, how you go from like a sample type all the way to sequencing. Sort of breaking it down, on the left is the extraction, extracting your sample. Then it goes through a few QC steps, the picogreen to quantify it and normalizing it to get this all ready for the actual library prep. Here in this slide is all condensed into just one step for library prep, but this will get, we will go into this in some of the next slides. Following that, you have your prepared samples, so you go through more quality control checks to make certain it's ready and able to be sequenced. Finally, qPCR in order to get a real accurate quantification of your sample, pulling them all together and then putting it on the sequencer. So some of my approach to doing this on the OT-2 is each of these little blocks would be a separate protocol, and they're all run on the same robot. Some of my approaches here for tackling the various library prep kits was to do batch sizes of about 24 samples. I found that's a good number. That's not too big or too small, and is just about the right workload for the robot setup that I have. It can do more samples, going up to maybe 48 samples, but beyond that, you begin to get diminishing returns. As we get into the robot, you'll see that we have a pair of multi 8-channel pipettes. So as it goes, the samples it will do is one column at a time. And as each column is treated separately during a step such as ampere cleanup, the more columns you have, the slower it takes. So above 48 samples, you're going to get sort of diminishing returns on the amount of time saved. Also, the deck space is a bit limited, so if you do go beyond 24 samples, you will have to be in close proximity to sort of reset some of the tip boxes. So why automate on the OT-2? Some perspectives is 96 samples is a minimum amount that needs to be automated because everything can be treated as a single block. There's larger robots for that. But for my purposes, I previously worked in a clinical lab in which we did small sample batches, but we did them repeatedly because sample turnaround time was quite important. So we wanted to be able to do small batches repeatedly and be flexible enough to change between 16 samples, 8 samples, or 24 samples. So this is a deeper look into the actual library prep. Basically, what library prep is taking your DNA molecules and then breaking them down into manageable chunks that can be read on the sequencer. The Illumina-based sequencers could read maybe 150 to 300 cycles. What that means is it could do 150 to 300 bases being read. If you have molecules longer than that, they'll sequence, but you might be missing some of the sequence in the middle of the molecule. And anything even longer might not even cluster accurately on the flow cell. So it's breaking up the DNA into manageable chunks. That's through a fragmentation. Fragmentation, depending on what technique you use, usually it leaves a random overhang. It just breaks the molecule randomly throughout the entire length. Then it's followed up by end repair, which blunts it. It trims off the overhang. And usually A-tailing, which adds an A base. So it leaves a region that the adapter could be ligated onto. As you see in the ligation, we're adding on an adapter. Usually this one contains a unique barcode. So later you're able to combine these samples together and run them on the sequencer in one batch. This is, well, you can multiplex multiple samples together. As you go and look at different library protocols, some protocols had different techniques in which they split up the adapter into 2 parts. Sometimes there's a full-length adapter, including the barcode as well as the regions needed for sequencing and binding to the flow cell. Other times they could split up the adapter, so actually the barcode comes in later during the amplification step. As you get deeper into NGS library prep, depending on what your project is, a lot of things could be switched around or replaced or changed. Maybe you're working with amplicons, so you don't need the fragmentation at the beginning. You're already designing regions to amplify that are already in the exact size you want. Sometimes you want to actually avoid, or you don't need the amplification. Either you start with a surplus of your sample DNA, so you don't need to amplify it, or maybe even you wanted to avoid the amplification bias or any bias that's introduced. So you would skip the PCR amplification and the final cleanup. Throughout all of this, quantification is important. This is a slide that I'll be referring to again later, generally showing on the left side the forms of quantification for a high concentration sample, and on the right side, something for low concentration. Throughout the course of your protocol, your samples will get smaller in volume, lower in concentration, and then you will want to pick out at the end just specific molecules, just the ones that will sequence, the ones with the P5, P7. So for all these different stages, there's a best fit form of quantification. At the beginning, you could do something like spectrophotometry, just looking at the absorbance. Let's say you're extracting samples out of saliva or blood or something messy. You have your extracted sample, you want to check the purity. So 260, 280 is pretty good. This method is pretty cheap and fast. You don't need any additional reagent kit, as long as you have a spectrophotometer. But this is only good to get a ballpark sense of what your DNA is. The numbers in blue would serve sort of as a guideline between a cutoff between if you want to do spectra or fluorescence. In the mid-range, which will probably be the bulk of your quantification methods, is fluorescence. This is generally like a pico-green kit. So it uses an intercalating dye. So it's a dye that fits between the grooves of a double-stranded nucleic acid, be it DNA or some other weird form of RNA. So this quantifies based on mass. So it's much more specific than doing it with spectrophotometry, because now it's ignoring anything that was also absorbing or obscuring the results. So now it's only the double-stranded nucleic material. This is a longer process. It is more reliable. It is more specific, but it does require a reagent kit as well as a fluorometer that can read it. And then finally is once you have your final prepared libraries diluted down, you want to do qPCR. So this is quantification based on a specific primer amplification, the P5, P7. And this quantifies based on quantity, not mass. This becomes kind of important if you have fragment sizes that maybe something went wrong in your protocol, your fragments are way too long or way too short. Fluorescence, if you have a bunch of short molecules or fewer long molecules, since it's based on mass, you wouldn't be able to tell it apart. So qPCR is based on just the quantity of molecules that will actually bind and amplify on the flow cell. qPCR, pretty accurate, but it is expensive, it is time-consuming, it is very specific, and it is very sensitive. So this is what is used at the very end to quantify your libraries before putting it on the sequencer. Now let's look at it on an OT-2. Here we started with 3 kits, Illumina DNA Prep, Roche HyperPrep, and NEB Ultra 2. For these 3 kits, we did the same starting material of 100 nanograms of lambda DNA and 8 replicates, 1 entire column. There is an important difference between these 2 types of kits. For Illumina, the DNA prep is tagmentation. So this is different from the library prep I mentioned earlier, which used fragmentation. Tagmentation uses Illumina's transposase to both fragment and tag the adapter simultaneously, so that's why they call it tagmentation. They've gone the extra mile to make it extra fancy by making it bead linked. So the transposase is linked to a bead. So not only does it fragment and tag it simultaneously, it will wash away anything that hasn't been tagmented. On the other side is HyperPrep and the NEB Next Ultra 2. These are similar to each other. This outside the steps of the protocol, they leave the fragmentation up to the user. So here in our particular case, we use the enzymatic fragmentation, but it's open-ended enough in this side so that you can use other forms of fragmentation, such as sonication or nebulization. Those 2 methods are low cost, but they're also very low throughput. They don't require additional reagents, maybe an instrument, but you'll be sitting there sonicating 1 sample at a time. Our approach was using the more automation-friendly enzymatic method, using an enzyme to do the fragmentation. This does require an additional ampere cleanup to get it ready for the library prep, but this was done up front. Both of these were done with 4 cycles of PCR amplification. This is what the OT-2 deck looked like throughout all my protocols. My approach is to have as many modules on the deck as possible so that there's no need to stop and switch decks, modules, or pipettes. So you can't see it here, but this is using a P20 multi and a P300 multi. So in the top left, you have a thermocycler. It is pretty bulky. It's an on-deck thermocycler from Opentrons, but it does take up positions 8 and 11, as you can see. Down in the bottom left is the magnet module. Down the bottom right is the temperature module. My approach was to have all the reagents sort of pre-aliquoted onto a 96-well plate and kept at 4 degrees throughout the course of the protocol. As you know, for the NGS protocol, I'm going to be adapting on barcodes. Each one's unique. Usually they come in 96 or even 3 to 4 well plates. And to avoid needing to stop the protocol and hunt and pick, I replated the barcodes, just the ones I was using, into this 96 well reagent plate so that when the barcodes are added, I just use a multi-channel. I don't need to hunt and pick during my protocol. So everything is prepared and ready to go. Here's what some of these components look like. Here's the OT-2, the P300 and P20 multi-channel pipettes. The magnetic module, it has a motor function, so it can engage and disengage, so the magnet goes up and down if you want to disengage the magnet. Temperature module, which heats and cools. And the thermocycler module that goes on the deck. For all the quantifications I was mentioning, here's the additional instruments and kits that were being used. So these forms of quantification are particular to each lab and particular to the scale or the size of the batches you'll be running. Sometimes you want a small little fluorometer. Sometimes you need something larger like a plate reader. And going quickly here, here are slides of some of the data from those 3 different NGS kits we did. Again, this is 100 nanograms of lambda DNA. On the left, we have batches of just 8 samples, so it would be one row of a plate going through the protocol. On the right is when we're doing sets of batches of 24, which would be 3 rows. So if I was doing, as I mentioned, the diminishing returns, if I was doing 48 samples, that would be like 3 columns. That would be like, 6 columns. If I were to do the ampere purification, I would have to wait. So adding more samples sort of draws out the protocol. What you want to look at here is the CV at the bottom, the coefficient of variation, which is the standard dev divided by the average. This is a shorthand way to sort of measure precision. What I wanted to see here is values below 20%. Here it is for the hyperprep. And here it is for NEB Next Ultra. Again, all of the CV values are below 20. Here are some traces from a fragment analyzer. Fragment analyzer is like a gel electrophoresis. It sort of measures, it sort of separates the molecules based on size. So the bottom axis is time or size. The top axis is fluorescence intensity. So what I wanted to see here is sort of uniform smears for all the samples. If you remember, the tagmentation and the fragmentation are random, so it's just cutting the DNA molecules randomly over time. So hopefully you get a smear within the right size. What I'm looking for here, based on what my sequencer was going to be, was about 250 to 300 base pair libraries. Using a fragment analyzer is a good way to check for if you have any adaptive dimers or to see if there's any additional humps that are of larger molecules. And should there be anything weird, you could do ampere size selection to sort of tighten up the size of the smear. Here's a table of the variety of sample types and batch sizes that we've done. Between 8 to 24, we've done lambda, human genomic DNA. The CVs for all of them, as you can see, are below 20%, and the fragment sizes are between the 250 to like 300 that I was looking for. Here's some pie charts just showing the sample read representation when I sequenced these libraries of 8 samples each. So we took each one, we sequenced it, and we wanted to see roughly the representation of each sample. It should be like 1/8 of a pie slice. This is with normalizing by hand. This is a summary of some of the run metrics of how the robot performed each run. So for each of them, breaking it down between 24 samples, 16 samples, or 8 samples, how many tips were required and how much time. P20s, usually one box, the P300s, multiple boxes. And then the column on the right side is when I used tip reusing. So this tip reusing is sort of a ease of use feature that a user could sort of program into the script to reuse tips. It takes advantage of the bot being able to keep track of these tips and their positions versus you can do it by hand, but it's an additional headache. This is particularly reusing tips only for the ethanol washes, or in the case of Illumina, for the tagmentation bead washes. I would switch out a deep well and use I would switch out a reservoir and use a deep well with individual wells for each tip, so they would keep clean and there would be no cross contamination. It's an ease of use feature. It reduces the amount of trash, the amount of tips used, and it reduces the need for my intervention to reset tips. If you see with the reusing tip boxes, at most I use only 3 boxes of the P300. For my deck setup, I can only fit 3 boxes of the 300, so it would use one whole deck setup. I wouldn't have to pause and reset things. Still, these protocols are not completely walk away. I would have to pause the protocol and it waits for user feedback to move a plate from a thermo unit to a magnet and then back again. But at least I don't have to stop in the middle of a wash to reset tip boxes. Now let's look at some of the quality control stuff. Quantification, normalization, and pooling. So after extraction, I would do the quality control checks. I would quantify it, but I would have to make a plate, put it to the plate reader, wait for those values as CSV, add it into my script for the normalization, so I would know how much I would need to normalize each step. So if they're looking for ways to sort of ease that transition between having to stop and move on to the next step. And then I like to make particular mention of all the manual pain points. I was a primary user in a clinical lab doing the library prep by hand, and for me, with plenty of experience, what are the most painful points that automation would be able to solve? Normalization, having to treat each sample with a single pipette, normalizing it to the concentration I want, going one by one by one. Barcode tracking, having to track which barcode was used where. In an ideal world, a computer or a LIMS system would keep track of that, but we do not live in an ideal world. Ampere purification, that was a major pain point. If you've done it before, it's easy, but if you do it 3 times a day, every day, it becomes quite tedious. So automation is a big help there. And qPCR, when we get into it, you'll see it's very sensitive and requires very careful pipetting and handling. The amount of samples you have, you're going to multiply it by multiple replicates, multiple dilutions. You could very quickly fill up a 3-4 well plate. Having to do that consistently from day to day, you could get a lot of variability. Sometimes if you don't have enough coffee in the morning, or maybe you had too much coffee and your pipetting is a little jittery. Here is normalization. I'll get into the details in a minute, but this is using a single tip to treat each sample, diluting it whatever volume is necessary to bring it to 100 nanograms per sample in order to start the library prep. All the various steps that go into the library prep, whether it's tagmentation or regular and repair. Once the library prep is done, you have your prepared sample, and so going through another round of QC, making certain that you have enough DNA in order to pool and put on the sequencer. You don't want to have excessive amounts of PCR cycles because that could lead to PCR duplicates. It sort of degrades the quality of the sample. Another round of normalization to make all the samples normalized and diluted down, ready for qPCR. This is diluting them down to about 1 nanogram per microliter. And here, doing qPCR setup on deck, this is using P20s on a multi-channel pipetting into a 384 well plate. For the qPCR, we're doing the dilutions on the robot. So if you have a 1 nanogram per microliter sample, that could approximately be 4 nanomolar. And so for the qPCR quantification, you dilute that down to by 1,000 to about 4 picomolar. And then once you have your final samples that are quantified, you have the data back from the qPCR, you know roughly what concentration they are, pulling them all together until you have maybe 1 or 2 tubes of your pooled libraries ready for sequencing. Once you have an established protocol, maybe even throw it on the sequencer, I get a sense of how your data looks. There's a few variables you can change here. The other steps, the picogreen, the normalization, the qPCR won't change, but within the library prep protocol, you can tinker it around depending on what your sample is and maybe even what sequencer you want to run it on. You could change a few things like fragmentation time, either longer or shorter, the amount of PCR cycles, maybe you're producing significantly more or you're not enough. Maybe you want a few more, a little bit more DNA so you have some breathing room, and clean up size selection. Maybe there's a few weird lumps in the fragment traces. You want to tighten up the size selection. So I mentioned here that we were doing normalization with a multi-channel, doing a single tip pickup. Here's some data from the normalizations. This is sort of a contrived random plate of samples. The CV is about 30%. Using the robot, trying to dilute it down to 6 nanograms per microliter. You see the result is like 5.8. The CV is low, is like 3%. Going from 30% CV to 3%. So even if your library prep, the CV is a little bit high, the normalization, if you do a normalization step, you could even that out. And on the column in the middle, that's diluting it down to 1 nanogram per microliter. So if I were to do this normally, I would set up my robots, I would do all the library prep, I would get my quantifications, I would stop, I would clean out the deck, I would switch out the multi-channels and put single channels in there so that I could do normalization. But I had a little workaround in which using the very flexible Python scripting, I'm able to pick up just a single tip using a multi-channel and use that to pipette into each well individually. And these are the results that I was getting from using a P300. This was just using a P300. You can use a P20, but it's not as smooth. There's a few snags. So this was just using a P300, being able to normalize all these columns. Here's that quantification slide again. So now we're going to talk a few things about qPCR. So your samples are normalized. They're diluted down to about 4 nanomolar, which is 1 nanogram per microliter, about 4 nanomolar. The aluminum quantification kits usually come with primers for P5, P7 to amplify those regions. There's like a representative library on the left. You see the green and the yellow. That's the P5, P7 regions. So if your molecule has that, it will bind to the flow cell and form a cluster. So that's what the qPCR is amplifying. And it's usually included with a dye a double-stranded dye. So anything that amplifies will fluoresce. So you don't need any necessary specific probes. The image on the right, this is to show that quantification of your final library is very important. Once you put it on the sequencer, it's akin to throwing a handful of seeds in like a yard. You don't want to be too crowded, but also you want enough coverage. So under clustering, the little circle on the left, that's where not enough landed. You will get molecule sequence, but you're not using sort of all the extra space on that flow cell. The one in the middle representing, that's good clustering. And the one on the right showing what happens when you overcluster. So if your quantification was off or you didn't bother or there was something wrong, you could be loading too much DNA. Those samples, those molecules will be binding, forming clusters, but they'll begin to overlap. And the image gets so messy that the algorithm can't pick out individual clusters and will have to throw the whole thing out. Here are some tips for your qPCR setup. So mechanically, doing the dilutions and adding it to a plate largely depends on the amount of samples and the plate size you have. If you have a 96 or 3D4 head qPCR instrument. So what we did in the OT-2 was a serial dilution. So you have 4 nanomolar libraries. You're diluting it down to 4 picomolar and running it against a bunch of standards. Usually it's from like 2 picomolar down to 0.002 picomolar, I believe. A serial dilution of 6 different concentrations. So after you have a new library made, you could run it on the qPCR and get an estimate of where it falls. If there's like too much variability, if it's varying by like 1,000-fold, something might have probably went wrong. So after a while, you can pare it down to standards of maybe just like 4 standards. Other tips, again, using 20 microliter volumes is good for, you'll need extra dead space and it's better for precision pipetting. You want to do technical replicates. So if you have a sample, you want to run it like 3 times. And you want to do a dilution replicate. If you have a sample, dilute it out by 10 so you could get like a broader coverage of how that sample performs at 2 different concentrations. The most helpful tip that I've had was to rerun previous libraries as controls. So if you have a library that ran decently well on a sequencer and you know the cluster density that you get from it, that is something you should hang on to and use it as a standard when you run your next qPCR for your future libraries. This is showing not only how well your individual sample performed, but also how well it performed on this particular instrument. So it's a handy thing to have. For your standards, you can only use them really 2 or 3 times before, especially the lower concentrations get a bit funky. Diluting your samples down in a tween solution. Diluting these samples down to about 4 picomolar leaves them so dilute that they might start sticking to the plastic labware. Know your fragment size if you have a fragment analyzer or if you could spare enough sample to run it on a gel. You could run a melt curve afterwards. It's a good way to check for dimers or any other weird size populations. And your sample should come out at around standard 2. These are the standards from 2 separate Illumina quantification kits. They were diluted and pipetted using the OT-2, and your sample should come out around the second standard, 2 picomolar or 1 picomolar. I want to switch gears here and talk about different forms of normalization. So I found this recently, and I thought it was pretty cool. This is an enzymatic method of normalization. So once I got to qPCR, doing all those samples, all those replicates, filling up a 3-day 4-well plate, thinking of other ways sort of to reduce the amount of pain points. There are other forms of normalization besides just stopping, quantifying, and then balancing them by hand. There are forms such as bead normalization, but I wanted to avoid doing yet another bead wash on the robot. So from IDT, they have something, it's the XGen Normalize module, which is an enzymatic way to normalize samples, which is automation friendly. So from my understanding of the normalized procedure is it swaps out your usual adapters and barcodes and uses the normalized primers, which have a region that a enzyme will bind to. So in the second step, first you replace the adapters. Now you're doing a normalized step, which adds a set amount of enzyme that will bind to those adapters. Anything excess of that won't be bound. Then you go through pooling. So you're pooling all whatever complexity you have, 12 samples, 8 samples, 24 samples, pool them all together. And then finally doing a reaction that will digest away anything that hasn't been bound by that enzyme, essentially trimming it all down to just the molecules that were bound by that enzyme. Since it was a set amount of enzyme added to each sample, now your sample is normalized. This is sort of a rearranged setup that the normalized module sort of uses. So the key thing here is that pooling all these samples comes before qPCR. Here's some results. On the left is a batch of 8 samples that was normalized by hand, and on the right is a batch that went straight into the Normalize module. So I didn't have to quantify them. I just followed the instructions of the normalized procedure. If you see at the bottom, the CV for doing it manually is 16%. And then on the right, using the normalize was 11%. According to their literature, they can get lower, but I was pretty happy seeing it being able to at least match what I could do by hand. So the key thing here was this was all pooled before the qPCR step. So here is a breakdown between those 2 different techniques, normalization by hand and the normalize. The one on the, so these are sort of a breakdown of the protocol metrics. The amount of time saved is about an hour, which may not be impressive, but if you see the text there on the left, if I wasn't pooling, I was pooling after qPCR, I have 24 samples. If I do 2 dilutions, if I have 3 replicates, now I'm doing 144 qPCR reactions. Whereas for the normalized, since everything is pooled before qPCR, I'm able to do just 6 reactions. To me, this is a good example of a technique that sort of can reconfigure the amount of samples and their setup in order to better fit an automation format. And there you go. I hope you got a good walkthrough of the OT-2 and what it's capable of how we approach automating the Illumina kit, the HyperPrep, and the NEB kit. I was quite happy with the data. The DNA prep samples had low CV, sequenced very well. And the biggest plus for me is it reduced a lot of hands-on time. So it automates all these time-consuming and repetitive steps, especially the magnetic bead cleanups. Automating this comprehensive NGS workflow on the OT-2 minimizes the risks during DNA library preparation while ensuring a quality DNA library prep for sequencing. Thank you for listening. That's me, Matthew Akana. Thank you.
Thank you, Matthew, for the really informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question button located on the far left of your screen. We'll answer as many of your questions as we have time for once again. Let's get started. Our first question here is really speaking to throughput. Can you normalize multiple plates at the same time on the OT-2?
Yeah. So if you saw from the deck slides, it has up to 11 usable deck spaces. So you can normalize, if you're only using like one tip per sample, you can normalize up to about like 5 plates. Note that it would not be simultaneously. It would be using a single channel, so it would be doing one sample after the other. So while it can fit that many plates, it will take a while.
Next question, do you need to have a specific DNA-specific internal standard control in order to quantify DNA with picogreen? So just elaborating on the fluorescent quantitation there.
Yeah, so it kind of depends on what instrument that you will be using to quantify to measure the fluorescence. What we usually do is that we get a picogreen kit from Thermo, like the high sensitivity DNA kit, and that comes with a control standard. It's like 100 nanograms per microliter, and then you just follow your suggestions if you need to dilute it to make like a series of dilutions, or if you just need a high point and a low point. What we've done it both ways. We have a qubit that requires like a high and a low standard, as well as doing it on just a generic plate reader in which we set up whatever dilution standards we want to run on our plate.
Got it. Can I design a protocol for a number of samples that are not divisible by 8?
Yeah. So if you've seen from the presentation, most of my batches were doing 24 samples, which is a nice set, 3 columns of 8. But if you're doing a number of samples that are less than 8, I would stick to using the single channel pipettes. If you're doing a set of samples that's like 12, what I would do was just pretty much omit the top and bottom rows. So I would have a row of 6 and a row of 6. Since I'll be using multi-channels, they'll just be treating everything simultaneously. So I would just omit those rows so I could fit in maybe 10 samples or 12 samples.
Very related to kind of protocol design, we have a question here on the tip reusing that you mentioned during the presentation. Is tip reusing now programmed into the GUI or only Python scripting? So please elaborate.
Right now, it is only in Python scripting. So the GUI is a good sort of introductory use for users new to setting up a protocol on the OT-2. But once you learn the Python, there's a lot more flexibility. So tip reusing, which I do for just the washes, is something that I was able to figure out. I'm not a programmer. I just started learning a few of the Python scripts. So it was something I wanted to set up, and then you could treat it separately. You can make it, I want to reuse tips and then put them back into like a designated tip box. Or I want to reuse tips, but then just to be safe, I want to make the pipette head move slower just in case there's any random droplet. So with like the trade-off is no, it's not on the GUI, but on Python, there's a lot more flexibility in how you could sort of use the tips.
Got it. Another question here. Are your negative control PCRs adequately clean when using the OT-2 for PCR setup? Do you need to have pre and post PCR robots, or has this been a problem in the past?
So far for me, most of my PCR on deck is using the on-deck thermocycler, and things have been pretty clean. Again, a lot of it is just general NGS library prep, which is pretty robust. If you work in a clinical lab, you do want to keep things sort of separate from pre and post PCR, especially for NGS, because a lot of the universal primers or adapters, there's fear that if they're circulating, something could amplify. So previous users would ask that question, and my suggestion is, yeah, you would have to sort of, to keep things CLIA compliant, you would break things up to pre and post PCR, like two separate reactions. The other option is the not listed, or it might have been listed, I kind of forget, is that the OT-2 does have a HEPA filter that could be fitted on top. The box itself of the OT-2 is not airtight. There is circulation, but you could put a HEPA filter on top to keep things cleaner on the inside if you worry about anything circulating.
Very helpful. So another question here, pretty specific. What should be the minimum concentration of bacterial genomic DNA for whole genome sequencing? Matthew, I'll lead the witness here. I'm hoping you can speak to kind of different ranges of DNA templates that you've used in your experience with the OT-2 library prep.
Speaking on that, most of my samples have been 100 to like 50 nanograms of like just extracted DNA. There's like a broad range, I would assume, of different applications, different things you're going to be investigating. So if you have a particular sort of protocol in mind, I would look into like their user suggestions. A lot of them do have sort of like a low input protocol that would sort of either introduce extra PCR, or if you're concerned about like PCR errors, it would do like PCR free. So it treats the protocol a little bit differently depending on how much input. Like if you could spare extra, you could use a standard input. But if you're concerned about like a very low amount, they have particular protocols. All of them, it's just changing a few steps on the existing OT-2 protocols. But they have stuff in particular if you have like a very low input. I would say, I think the general cutoff I've seen is like around below 10 nanograms of DNA. You would use like a low input protocol.
Absolutely. What is the Python code to pick up a single tip using a multi-channel pipette head? Very specific to the Python API here, but how have you accomplished this in library prep protocols?
There's two different ways. One again, I'm mostly new to Python, but I was able to sort of jury rig a tip pickup by just setting a bit of physical offsets. So you have to make certain the deck is clear. But a better suggestion, I believe there's a technical note we have from the apps engineering team about how to sort of do a single tip pickup. The requirement is you have to be a little bit familiar with the Python protocol and sort of how to make a few changes. But there's like some variables within the tip pickup factor to sort of make it a more gentle pickup so you don't crush anything. So I believe
Absolutely.
we can share that. I'll have to check.
Yeah, definitely refer to our support documentation on opentrons.com for more detailed reference to the code there. A question about dead volume. How do you overcome some of the dead volume with these very sensitive library prep workflows?
Um, the way I've done it is if I can get away with everything being pre-aliquoted into like a strip tube, I would do it that way. When I do it that way, I check. Sometimes they sell the protocol kits in like 24 samples, so I would measure how much do they provide. So therefore, how much extra dead space do they allow? Some are more finicky, and so what I would do is I would put like a foil seal on top of the well, so there's no evaporation over the course of the protocols. When it goes to that reagent, it should still be there. Some of the foil seals, I'm trying to look for one, but most of the foil seals that I've used are like puncturable, so I could put a foil seal on there, not worry too much about anything. The pipette would go in there, poke a hole, actually like poke a hole, widen the hole just a bit, and then take the reagent out.
Thank you. We have a question here about a low yielding library prep that someone is optimizing, even manually, potentially. So, is there anything that you can speak to specifically about troubleshooting a low yielding kind of library prep workflow?
There's a lot of steps. The ones that I think are the biggest culprit, if you have a protocol and it works fine manually versus putting on the robot, so it could come down to the way the robot handles it. The main culprits that I've seen from my experience are either the ampere cleanup with the mag beads or some of the reagent pipetting. For the ampere, the usual thing is either the bead settling time, either you have like a large volume and it takes forever for the beads to settle. And so when you move the supernate, you usually could get like bead carryover into the waste. So you're losing DNA. That or not removing all the ethanol. Maybe the tips are misaligned or you're not getting all the way to the bottom. So there's still ethanol in there when you do the resuspension. The other culprit could be sort of the reagent mixing of all the sort of reagent enzymatic steps. Usually it's ligation, the ligase, or sometimes the fragmentation. Those buffers could be extra thick or viscous. And so compared to do it just manually, yeah, you could do a lot of physical pipetting and mixing. But with a robot, you would probably want to increase the pipette mixing step so that the ligase buffer is like fully mixed in with the reagent sample.
Yeah, a bit of a root cause analysis there. While you're mentioning kind of pipetting efficiency, are there any recommendations for quick and effective pipette mixing, especially on full plates of libraries prepared?
Um, from my presentation, I'm sort of mentioning that, yeah, you might have diminishing returns if you want to do 32 samples, 40 samples, because the multi-channel pipette head is going to have to go in and physically mix each column one by one. There are options if you want to push it to like a 48 samples or even 96 samples. Instead of using the multi-channel mixing it one by one, would be just to stop the protocol and move it onto a separate shaker so it could shake the entire thing at once, and then maybe do a spin if you're worried about it being shaken too much. I do believe we have a heater shaker coming for the OT-2, so that will be an on-deck shaker that you could do all your reagent addition. Let's say you're doing an ampere, you add the ampere beads, and it will shake the entire 96 plate at once. So you don't have to go in manually and mix each thing one by one. Because if you're doing things one by one, maybe one column has to sit a lot longer while all the other columns get washed. So you might get some unevenness, whereas the heater shaker will shake the entire thing.
We've got some more questions coming in related to bead cleanup on the OT2, as you're referring to the ampere. cleanup. Have you noticed any over drying of beads before the samples are able to be resuspended? Great question here.
I personally haven't. I have noticed under drying. Just switching protocols and plates, sometimes it would under dry compared to over dry. Over dry, if that happens, yeah, you would just reduce the your pauses or the weights it has for evaporation. What it turned out to be for a previous case was that sometimes the samples were in a 200 microliter plate versus a 1 mil deep well plate. So the 1 mil deep well plate had a lot less air circulation, so it didn't evaporate as fast as the shorter plate. So for that, even though the timing steps were the same and the volumes were the same, there was a lot more of the ethanol that didn't get evaporated away. And if you do have cases, or if you're worried about overdrying, would be increasing sort of the resuspension steps. The ones that I have physically locate the tip against the surface of the bead pellet, so that the washing is a little bit more vigorous than just pipetting into like the center of the well.
Very helpful. One question, are there plans for, or are there already webinars for more in-depth informational sessions on the Python protocol development? I'll take this one. Matthew, if there are Python questions, please check out the webinar section of our website, opentrons.com. There are previous webinars to reference on the Python API with our really talented applications engineering team leading that. So please check those out. There will definitely be more in the future because there's quite a bit of interest in that, as I can tell from the questions we're getting today. I have one more question for you, Matthew, here as we're coming up at time. Can the OT-2 perform any other NGS steps such as extraction?
Yeah, so if you remember my presentation slide of all the steps, one after the other, extraction is the very first one. I've able to incorporate a version that works for me with the setup that I like. All the extractions are basic sort of bead extraction kits. The one condition is that it does require a bit more of the deck setup because the amount of sample and the amount of beads and the amount of washes leads to a lot more liquid waste. So you have to account for, I don't want to just dump the liquid into like the trash receptacle. So I have like deep well plates just for all the liquid waste. I just have like one more note is thank you for going through all the protocol. My big suggestion is to sort of follow once you have a kit or protocol in mind, is to follow their recommendations. So the slides the workflow I presented was sort of like a generic basic template for a lot of people to get started. But sometimes the manufacturer might require something is like more optimized for sonication instead of the fragmentation by enzymatic means. Or sometimes based on your results, every lab is different. Maybe you need like tighter fragmentation sizes, so you can include like the ampere bead size selection. So that would be like a whole step unto itself because it uses so many tips. And that could come like early on, it could come like later in the protocol, it could come like after the library prep. So one of the benefits of all these protocols and stuff that I've experienced is very flexible. I'm able to sort of slot around different steps, include or skip over certain steps within my protocols in order to get the sample sizes or specificity that I want.
Thank you, Matthew, for speaking to the flexibility of the platform. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by the speaker via the contact information provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.